I'm Duncan Baird. I head up the telomere laboratory at the medical school. We're currently between CRUK programme grants, and so we've spent the time during lockdown finalising the analysis of the data that we generated during our last programme grant, writing up the various manuscripts, writing grants, and preparing for the next 5-year programme grant that we have. We're hoping that we can be back in the laboratory soon, start generating new data.
Hi, I'm Katie, a first-year PhD student in the Tudor Group funded by the School of Medicine. I'm a bioinformatician looking at genetic markers associated with toxicities to colorectal cancer treatment. During lockdown, I have run genome-wide association studies using over 5 million genetic markers, which have identified one significant locus, 5 validated or suggestive significant loci, and secondly, 68 low-frequency loci. I'm currently using Bayesian file mapping with over 8,000 annotations to identify plausible biological candidates. I've also been learning some machine learning methods, which I hope to use on our datasets in the near future.
Hi, I'm Chris. I'm a first-year PhD student, bioinformatician working in the Cheadle Group. I'm funded by Tenebus Cancer Care, and during lockdown, I performed a genome-wide association study of survival time in colorectal cancer using 2 million common genetic mutations across the whole genome. I found 17 independent loci showing a suggestive association with survival. I'm now validating these in an external cohort and performing downstream gene analyses to further understand what's going on there. I've also been working on developing machine learning skills to build prognostic models for colorectal cancer using clinical and genetic data.
Hello, my name is Hannah West and I'm a research fellow in the Division of Cancer and Genetics at Cardiff University. During lockdown, I've been really busy analysing genetic data to try to find new genes that could be predisposing people to bowel polyposis. And people with bowel polyposis are at a much greater risk of developing bowel cancer. I've also been busy looking at genetic data from cancer samples to try to spot mutational signatures across these samples.
Hi, I'm Victoria, a third-year PhD student within the Cheadle Group. I'm a bioinformatician and biostatistician. During lockdown, I performed an analysis of over 5 million unique points across the human genome, searching for markers that may predict how late-stage colorectal cancer patients respond to chemotherapy. I have found 8 unique loci that may be associated with this response, one of which is supported by a significant change in how long patients survive on this treatment. I have also been involved in facilitating a virtual international meeting with colleagues as far away as New Zealand and Hungary that happens every few months. I've also presented my own work at one of these meetings, so I've been keeping very busy.